Eurohyp-1 Biomarker Study Protocol. Blood collection. Take the material for blood collection from the patient sample kit. 1 safety tube holder, 2 serum tubes, 2 plasma tubes, and 1 RNA tube contained in the blood collection kit. Please check the labels for patient code and sample time in order to be sure you are using the correct tubes. Take the safety tube holder and remove the needle cap. Perform the venipuncture according to safety and standard procedures. Take the serum tube and press it to the bottom of the holder to start collecting blood. Please be careful to avoid hemolysis during blood collection. Please be careful to avoid hemolysis during mixing. Take the plasma tube and press it to the bottom of the holder to start collecting blood. Take the Tempus RNA tube and press it to the bottom of the holder to start collecting blood. Blood processing. Place the RNA tube in the RNA storage rack and store in the freezer at -80 degrees Celsius. Keep serum and plasma tubes in the fridge at 4 degrees Celsius for at least 30 minutes to avoid blood to clot in the serum tube. Within 4 hours of blood collection, centrifuge the serum and plasma tubes at 1500 for 15 minutes at 4 degrees Celsius. Please try to balance the tubes in the centrifuge, as weight imbalance could cause the centrifuge rotor to break. After centrifugation, the tubes look like this. Serum, separation gel, and blood cell rests in the serum tube. Plasma, a thin layer called buffy coat, and red blood cells in the plasma tube. The buffy coat is the only component that contains genetic material, DNA and RNA. We only can obtain it from EDTA tubes. Blood allocating. Take the patient storage rack, the patient sample rack, and the material from the blood allocating kit. 3 plastic pipettes, 1 yellow cap strip, 1 blue cap strip, and 1 red cap strip. Fill the microtubes in the rack according to patient sample distribution. Please check the microtube labels for patient code, sample time, sample type to ensure the use of the correct material. By using a plastic pipette, draw out and transfer the serum to each of the 7 serum microtubes, filling them more or less to the upper border of the label. That is approximately 0.5 milliliters. Please be careful to avoid drawing out the separation gel. Cap the 7 microtubes with the yellow cap strip by pressing it to the tube. Remove the plasma tube cap. By using plastic pipettes, slowly draw out the plasma from the top and transfer it to each of the 7 plasma microtubes, filling them more or less to the upper border of the label. Cap the 7 microtubes with the blue cap strip by pressing it to the tube. Transfer the filled microtubes from the patient's sample rack to the patient's storage rack by using the cap strip. Tear the strip to release the caps. For the DNA aliquot, take the plasma tubes. By using a plastic pipette, draw out and transfer the buffy coat to each of the 2 DNA microtubes, filling them more or less to the upper border of the label, till approximately 0.5 milliliters. Cap the 2 microtubes with the red cap strip by pressing it to the tube. Transfer the filled microtubes from the patient sample rack to the patient storage rack by using the cap. Remember to store the remaining caps for serum, plasma and DNA microtubes in the reserve material kit. Sample storage protocol. Store the patient sample rack in the freezer at -80 degrees Celsius until further shipping to the Eurohyp-1 biobank.